Good afternoon. Today I'm going to discuss about a new chapter that is microbial cell culture. Okay, so microbial cell culture, it is a technique in which we are going to utilize the bacterials, that is for the production of different useful compound like antibiotics, then production of protein biomass like single cell proteins inside the microbial cell culture media. So we do that activity inside the lab. This is a lab based experimental chapter that we are going to prepare the media, that is the food of bacteria, that contain the carbon source, the nitrogen source, then the other vitamins, minerals, then so many nutrients that facilitate the growth of that bacteria. So we are going to prepare the food of that bacteria after preparation, mixing with the different inorganic source, carbon source, vitamins, minerals inside the water. Then we maintain the temperature and pH, especially pH, because each bacteria will grow at particular pH. So we have to maintain the pH by pH meter or pH spray, either with the help of dilute H hydrochloric acid or dilute sodium hydroxide. If the requirement of acidic media, then we add drop by drop dilute HCl. If the requirement of basic media, then we add drop by drop dilute sodium hydroxide, dilute solution of sodium hydroxide. So these are the first step for preparation of media for the growth of bacteria, because bacteria cannot grow without media. They are consuming the media from the nutrient from the media. So there are so many culture techniques before going to utilize by that media. We should do the process of autoclave. Autoclaving, I have already discussed this thing in class 11. Means after preparation of the media inside the water, after putting the energy source nutrient, carbon source nutrient, nitrogen source nutrient, oxygen source nutrient, then minerals, micronutrient, macronutrient, means this media is common structure, framework for each type of media for bacterial culture, media for fungus culture, media for algal culture, or media for plant tissue culture, or media for animal cell culture. Something is common. So mainly there are three type of media. One is synthetic media. This is the word is indicating synthetic media. This means we are going to prepare the media with purely the chemical substance. Means we are utilizing only man-made chemicals for the preparation of the media. Then we are going to prepare the natural media, semi-synthetic media. In case of semi-synthetic media, there is a mixture of both natural source as well as synthetic chemicals that can be synthesized in the lab, in the chemistry lab, in the chemical engineering lab, that is synthesized. Means we are going to mix both the type of source of nutrient. One is from natural source as well as from artificial source, that is man-made source, that is known as semi-synthetic media. So try to remember the example of semi-synthetic media, that is the beef extract, yeast extract, then casein digest. So we can prepare that media, that there is a combination of both natural source as well as the chemical or synthetic source. So we do the processing of the media. Before processing of the media, we should consider some points that is given in your books. That is, what is the carbon source of that media, what is the nitrogen source of that media for bacterial culture, media for microbial culture. What is the carbon source? You can get the carbon from a starch, cane, molasses, it's sugar cane juice, glucose, that contain the carbon. We have already discussed the structure of glucose, fructose, sucrose, lactose, where there is aldehyde or ketone groups, or you will find only carbon, hydrogen, and oxygen. So generally, carbon source are starch, cereals, grain, molasses. They, while mixing in the media along with water, because water is a universal solvent. Next is nitrogen source. The need of nitrogen for making protein formation, for making polypeptide formation, for the building block of the body of the microbial cell culture, microbial body. So ammonium salt as a nitrogen source, urea as a nitrogen source, corn steep liquor as a nitrogen source, and slaughterhouse waste means the body where there is a killing of animals, whatever the rest left after selling the flesh, then whatever the material left, that can be utilized for preparation of media. That is the good source of nitrogen. Then trace element, you know these things. Trace element, that is the act as cofactor for different metabolic pathway, because each metabolic pathway require what? That is the enzyme. And enzyme are activated by micronutrient like iron, copper, zinc, manganese, molybdenum in a small quantity to activate the enzyme, just act as cofactor. So we must add a trace element in very little amount. Then next factor, that is the growth factor. What is the growth factor? Means amino acid, vitamins. These are the growth factor you must add when we are going to prepare the media, because that is required for the multiplication of that bacteria, the multiplication of that fungus, and multiplication of that any microbes, that is required. So next component of the media is anti-foaming agent. What do you mean by anti-foaming agent? You know, the chemical that prevent the formation of froth or foam. Means if the bacteria is multiplying in any liquid, that is the media, and they are respiring, they are releasing CO2, and during respiration that CO2 will create a foam like a structure in the media, and it also increase the temperature. So that foam prevent the growth of that bacteria, that fungus. So we use generally some chemicals that prevent the formation of foam, so that they know stopping the growth of that bacteria as per our requirement. So what are the anti-foaming agent? That is olive oil. Generally, so 10 to 20 times this question has been asked in your CBSE exam. What is anti-foaming agent? A chemical that prevent the formation of foam, froth. I think you have read the froth flotation method in chemistry. So formation of foam, झाग जिसको बोलते हैं, that it should not form inside the media, liquid media that is going to utilize for growth of bacteria, because it will create interference, it will create a disturbance for the growth of that bacteria. So what chemical you can utilize? You can utilize fatty acid such as olive oil or sunflower oil. Silicones are also used as anti-foaming agent. So you have to remember the terminology and what is anti-foaming agent, what are example of anti-foaming agents, and what is the advantage of using that anti-foaming agent inside the microbial culture. Okay, and what is the function of that trace element, that is micronutrient. You know, micro and macro, you have studied these things in chemistry as well as biology also. So micro, micro element that act as cofactor, activator for different enzymes, and macronutrients. I have already discussed these things in plant tissue culture. Macronutrients, you know, carbon, CHONSPK, that is the single letter symbol of different uh, elements. C for carbon, H for hydrogen, CHO for oxygen, N for nitrogen, S for sulfur, P for phosphorus, K for potassium. So these are the nutrient, that is the elements that is required in huge quantity for making body part of the any microbes, any living organisms. So that is come in the category of macronutrients. So try to remember these things, what is happening in case of uh, preparation of the media. What are the requirement, what are the source of nutrient? Carbon source, nitrogen source, okay, and sorry, and the protein source, then vitamin source. So you try to remember these things, what are the source of nutrient in the media. And you know, energy source, that is the carbohydrate, that is the lipids, the protein, that is the main source that provides the energy for the growth of microbes, because every organism requires the energy for its growth and multiplication, and source is only carbohydrate and lipids and protein in the culture media. Next, you know, water. Water is a universal solvent just to mix all the nutrient, all the components of media. So that maintain the pH, that is, that facilitate and provide the availability of the nutrient that is utilized by bacteria. So it also maintain the temperature, heating and cooling. So clean water of the constant consumption is therefore requisite for large scale cultivation of microbes. So water, you know, due to high dielectric constant, it is a universal solvent. It will maintain the temperature of the body, microbial body, and supply the nutrients to microbial body. So these are the universal solvents that can be utilized by the bacteria or any microbes. Next is culture procedure. What are the culture procedure? You can proceed for multiplication of bacteria, for culturing of bacteria. First, sterilization. Sterilization is the first and foremost technique for cell culture of microbes, or cell culture of plant cell or animal cell. So we should follow a strict sterilization procedure before starting of culturing of any microbial cell culture. Means ideal condition is that means temperature is 121 degrees Celsius for 15 to 20 minutes and the pressure is 15 pound. I have revised these things in class 11 so many times. That is the ideal condition for autoclaving. Without autoclaving, we cannot proceed for microbial cell culture. So this is the most important steps in microbial cell culture for the production of antibiotic in the pure form or the for production of any protein in the pure form through microbes. Microbe means either we can utilize the bacteria or fungus. You know fungus, that is penicillium, that is used for production of penicillin. Antibiotics, that is the fungus, and there are so many bacteria that can use for the production of antibiotics, production of protein, production of so many useful compounds, hormones. So concept of molecular farming, I have already discussed these things. Farming means production of any novel useful protein or compound that have medicinal importance. We can adopt any bacterial cell for large scale production of any protein, that is known as molecular farming. So first and foremost technique we have to follow, that is sterilization, means complete eradication of microbes, unwanted microbes, harmful microbes that can destroy, that can degrade your desirable protein, desirable antibiotics. Means it can, the bacteria free condition of any glassware, any media, so that we can grow only pure, only particular bacteria inside that media. So whatever you are for this purpose, that is the cell culture, we should follow the sterilization. So there are so many methods of sterilization. That is, first is autoclaving. What first is autoclaving. In this case, ideal condition I have already discussed. That is 121 degrees Celsius for 15 foot per square inch pressure per square, 15 pound per square inch psi per square inch pressure, 15 to 20 minutes. That duration will vary as per our volume, that which we are going to autoclaves. So if we are increasing the volume from 100 to 200, 300, 400, 500, 600, 1000 ml, then further, then we have to increase the duration of time also. After autoclaving, we can open these things in laminar air. Otherwise, again there will be a same contamination occur from the environment. So we do all the procedure in aseptic environment. That is the environment where there is no bacterial spore in the environment, and we can do our experiment successfully without any contamination. So that is the sterilization, means hot air sterilization, moist heat sterilization, surface sterilization. So first is autoclaving, hot air sterilization, moist heat sterilization. Hot air sterilization means we are going to autoclave the glasswares. Whatever the glassware we are going to utilize for our experiment, we do the process of autoclaving sterilization through hot air sterilization. Then moist heat sterilization, we can do that sterilization at with the specific heat of moisture, moisture to kill the microbes. And next, that I already discussed the autoclaving. We can also do the surface sterilization, surface killing of microbial culture, if it is present on the table, on our inside the laminar airflow, or where we are doing the experiment or culture room also. We can also do the fumigation, AMNO4, so that if there is a spore present in the culture room where the bacteria is going to multiply, or the lab environment where bacteria can infect, the bacteria in the atmosphere can infect our experiment, affect, contaminate our experiment. So we fumigate the whole environment so that there is no chance of infection, there is no chance of contamination. Next, aeration and mixing. We can provide the proper air, so that oxygen, so that bacteria can multiply properly inside the fermenter. So try to know what are the equipment for microbial cell culture. What are the equipment for microbial cell culture? So you know, Beffel flask. It is a conical flask. You know, this is the Beffel flask. That is the conical flask. And next, in which we are going to prepare the media. Next, you know, this is the fermenter, bioreactor. You know, this is the diagram of fermenter. So this is the most important instrument in microbial cell culture, where we clean the fermenter after cleaning, after rinsing, so that there is no chance of contamination. We remove all the contaminants, we rinse it with the water, hot water, and then we pour the media. And then after pouring the media, 3/4 portion, we put the particular bacterial culture as per the requirement for the multiplication of that bacteria or microbes inside that fermenter. So just this is the instrument in which there is a proper multiplication of that bacteria, that fungus can occur, so that we can produce huge quantity of protein. Protein can be hormones, protein can be antibiotics, protein can be used as food for animals or humans. So after preparation of the media for the bacteria, we pour this media inside that. Means put the media inside that. Okay. Then we put that bacteria, and you try to know what is the design. Just try to know what is the design of that fermenter. So you see there is a blue color, that is the water cooling jackets. You know, when the bacteria will multiply, they increase the temperature of the environment inside that fermenter. So what happens if the raise of the temperature, raising of the temperature, it can affect the growth of that bacteria. So first, there should be a proper facility of water cooling jacket that maintain the temperature due to respiration by bacteria. In the center, you will find, in the center of that fermenter, you will find what? What you will find? You will find a motor and impeller. So motor, a rod-like structure through which a motor is attached on the top, and then you will find the impeller. Impeller give the movement of media, so that there is a proper mixing of that nutrient, proper aeration occur in that media, and bacteria can properly multiply. Because there is no movement, bacteria cannot multiply. So motor will rotate the impeller. Then what when you will do this after pouring the bacteria inside that media, then there is a system for checking the. So just try to no, there should be system for checking the pH. Okay, so and oxygen condition. So we maintain the temperature, we maintain the pH, we maintain the oxygen, pH, as well as other environmental condition, that is relative humidity inside the fermenter. When the completion of fermentation occur, then we stop the process of fermentation, switch off the fermenter, and then we collect whatever the material is processed, utilized, the nutrient is utilized by that bacteria inside that media, and that bacteria transform the nutrient into proper compound, that it can be a protein can be enzyme, or protein can be or antibiotics, or protein can be or hormones. So try to know what happens during fermentation. After completion of fermentation, in sufficient oxygen condition, how the bacteria get oxygen? Bacteria get oxygen through this asperger, sterile air, that is supplied from the bottom side, and oxygen is going to release inside the media, and that impeller mix the oxygen, so that every bacteria present in that media, uh, that inside the fermenter, gets sufficient amount of oxygen and do the process of fermentation. Okay, so you should you have to draw the diagram of fermenter. What is the basic design of fermenter? Every should know the basic design of fermenter. So today I'm going to give homework, that you have to prepare, draw the diagram of fermenter, and draw the diagram of different instrument, this different equipment, and different glassware that is going to utilize for microbial cell culture. Only then you will be familiar to what happens, how it happens in microbial cell culture. So you have to prepare the project, then homework. I'm going to give you homework, and you must complete that one. Okay, so today is also your exam. I think there is other exam also, so just go for uh, this homework as well as project. Okay.